Hi everyone. I was so excited to get into our unit that I missed a few slides and I had wanted to review this with you. So I've just created this short screencast to bring everyone up to date. For many of the tissues we will be talking about, it is important to understand how the cells stay together and how they pass nutrients, etc., along to each other. I'm going to briefly review the cell junctions of same cell types and cell junctions of different cell types. Remember, we reviewed the four basic tissue types. Well, the cells in each type are from the same family, and they attach to each other differently than they would attach to cells from another type or another family. So we'll be looking at that. So for most of the cells of the body, they're packed very closely together to form tissues and organs. But regardless of whether they're the same cell type or different cell types, there's always a space between the opposing cell membranes. If you remember when we were looking at that little video at the beginning of class, we could see that outside of the cell space that things will float around in. We call that the extracellular material. And it's there that it provides a pathway for the diffusion of substances between cells. So we're going to look at some of these cell junctions individually. Here's a diagram of, and this was taken out of an anatomy and physiology textbook. So you'll be able to find this in any of your textbooks, A&P textbooks. Usually something that is covered at the very beginning of A&P. And so we'll look at a couple of these different cells. So when you see this pink cell, that would be a typical epithelial cell. You can see the cube shape. This is the cilia or on the surface. So we're looking at basically half of one, half of, or a quarter of another, and a quarter of another. So it's the space between these two cell walls that we'd look at on the long sides. Close up, we'll see some of these and look at these closer in subsequent slides. Then when we look at the purple color here, that is a different tissue type. That is our connective tissue. So the bottom of the epithelial cell rests on that and connects to the underlying connective tissue. So we'll look at how that type of tissue, those two tissues connect together. So we'll start with the tight junction. This is known as the zonula occludens. This is when the outer layer of adjacent cells can appear to be fused but are not. It forms basically an impermeable membrane, and this causes any molecules that are trying to get through, they have to go around and find a different path to get through from one cell membrane into the other cell. So it's kind of like the teeth of a zipper. When you do up your zipper, they interlock and sort of connect together. So we see these in our odontoblasts that we'll be talking about, and with regards to teeth, and then in the lumen of the intestines as well. The second part of the tight junction is the zonula adherens. And this is basically the two cell walls are held slightly apart by a ground substance of medium electron density. So it's an amorphous ground substance. Amorphous just means structureless. There are tiny little tonofilaments found embedded into the cell's cytoplasm, so inside the cell. And so these little filaments, and we talked about tonofilaments yesterday, will kind of stand on end in that area. We have these these junctions are found in epithelial cells, which we'll be talking about a lot, fibroblasts, which also contributes to our lesson, and smooth muscles, as well as the heart muscle. Tight junctions are made up of both the zonula occludens and zonula adherens. So it's a combination of those two things that are called a tight junction. So a gap junction is the next junction that we'll talk about. It's very similar to a tight junction in that things can't pass between, they have to be directed. But what happens with a gap junction is there are these little tiny channels. They're hexagonal in shape, a little cluster of these small little channels that are in a hexagon pattern, and they will pass between the two cell membranes. So they act like little portals that allow the cytoplasm of one cell to flow into the cytoplasm of the next cell through these little hallways or channels. Of course, the size of the hexagonal subunit is going to restrict what can pass in and out of there. We see these in our heart muscle cells, smooth muscles surrounding blood vessels, our intestinal tract, those areas where you want the information to pass fairly quickly. So there's a picture of it on this side. Here we have the little subunits here that pass from the inside of one cell to the inside of the other. Then we also have these little, almost like Velcro tabs. These are seen on the long side of the cell walls. So I'll show you in the next diagram, the side rather than along the bottom. And it's a small gap with between the cells with a discrete disk of matter on the cytoplasmic surfaces of each cell. So on the inside of the cells rather than on the cell wall, there is this material, and it's moderate electron density, and basically they are attracted to each other, so they form like these little spot welds. And off of that, we will see these tunnel filaments that will stand up, basically, so you can see them on the side walls. This is a picture of the desmosome here. So these are the tunnel filaments. That's the discrete disc or plaque. And then this is the adhesion material of the two cell walls. So here's an example. Oh, it's on the side of the cell. When we have two different cell types connecting to each other, for example, an epithelial cell connecting to a connective tissue, we only have half of one of those spot welds. And this would be found on the basal surface, the bottom surface of one cell, which would be the epithelial cell. And it forms a firm adhesion of one tissue type to the other. So we see this in tissues that are prone to disruption, like our skin. And so we're firmly attached to the underlying connective tissue, the oral mucosa, the esophagus, vagina. In our oral cavity, again, the sulcus, or the epithelial attachment to the tooth surface, is stuck to the tooth through these hemidesmosomes. So this is a hemidesmosome found. So there's the basal surface of our epithelial cell, and there's the underlying connective tissue. Here's a larger picture of it over here. And we have these little half spot welds. So in other words, there isn't one on the connective tissue side. It's just on the inside of the epithelial cell. We have the tunnel filaments that will stand up and be evident from the electron dense material that helps to hold things together. Which now brings us to the basal lamina. The basal lamina is an area between the basal surface of one tissue type and the surface of the different tissue types. So this is how basically we have a structure that holds the two tissue types together. So we see this as an acellular structure, meaning there is no structure to it. It is a fine matrix of fibers and mucopolysaccharides that they believe is a secretion from the epithelial cell. And this structure is not oriented in any particular way. It's just, there's no cells in it. It's just a fine matrix of fibers. When we look at it under an electron microscope, it's seen as two layers. There's a dark layer and a light layer. So it's been given these two names, lamina lucida, meaning light, and lamina densa, meaning dense. Next to this, in the connective tissue, you would see anchoring fibers called the reticular lamina that will aid in attachment of the epithelium to our connective tissue. So this is a picture of that. I apologize, it's not a great picture of it, but here we have the lamina lucida, the light layer, and lamina densa, and then we have the anchoring fibers down in the connective tissue. So there's always this basal lamina between two different tissue types. We used to call it the basal or basement membrane, so it had two names, but now we call it the basal lamina. So you may see it as both in some of your textbooks. The main functions of the basal lamina is to provide support and elasticity to the tissues. It creates a strong connection between the basal cell layer of epithelial cells and the underlying connective tissues. It acts as a sieve to filter and modify and control molecules, the size of molecules that can pass through. It has some important functions. So I hope that this was a good enough review for you. There will be questions about this on the test, Unit 1 and 2. So if you have any questions, please let me know. Okay, thank you for your time.